The following is a selected video from masterthecontent.com, where you will find an extensive video library of lectures for a variety of standardized admission tests. We offer over 600 hours of detailed video lectures for a multitude of standardized tests. Use our interactive in-lecture table of contents to find specific topics of interest. Work through numerous in-lecture examples to help you internalize concepts. To learn more, visit masterthecontent.com. Your career, our passion.
Talk about or expound on these transcription factors that we're talking about. Okay, so transcription factors in eukaryotic cells. Without transcription factors, transcription would not take place. So what this slide does, it shows you a few important transcription factors and how they actually perform their function. For instance, we have transcription factors called activator proteins. And now what an activator protein does is that it will bind to a segment of DNA we call an enhancer. So an activator protein will bind to a segment of DNA called an enhancer. And once it does this, it will cause that segment or that portion of DNA to bend and bring it in close association with a promoter. Now, as you can see, we have activated proteins right here, associated or in unison or attaching themselves to enhancers right here. And what happens is that tandem, that combination of activator proteins and enhancers actually allows them or bends the DNA molecule to bring it closer to the promoter. Now, it doesn't matter if these enhancers are several thousands of nucleotides downstream away from the promoter sequence of target. As you can see, that DNA strand just bends around such that it brings those transcription factors into close contact with the promoter. Now, the assembly of these activator proteins and these enhancers makes it much easier for RNA polymerase to attach to the promoter and begin the process of transcription. So factors like enhancers and activator proteins basically assist or help RNA polymerase to bind to the promoter so it can begin the process of transcription. So this process, or what I'm talking about, is a process that promotes transcription. It's important to know that transcription can also be regulated negatively, or it can be regulated in the negative sense. We have other proteins that we call insulators. Now, what an insulator does is that it will bind to another protein that we call CTCF, or a protein called CTC binding factor. I'm talking about this protein right here. And once that insulator sequence is bound to that protein, CTCF, it can then block the action of enhancers. So it can prevent enhancers from actually associating with the promoter region. So that's one way through which transcription can be negatively regulated or negatively controlled or slowed, if you like. Now, the activity of these insulators is also regulated. So even the mechanism that slows down or that inhibits replication can also be regulated by yet another process. So what happens is that if the cytosine nucleotides in an insulator sequence are methylated, and what I mean by methylation is basically the addition of a methyl group to their cytosine nucleotides or to their nucleotides that have cytosine, that can prevent those insulators from blocking enhancer action or enhancer activity. So as you can see, we have so many different mechanisms, so many different transcription factors, all are working in different ways, all doing antagonistic things with the role of regulating the process of transcription. We have, for instance, activator proteins and enhancers that promote transcription, and we have insulators that actually block the activity of enhancers and inhibit transmission. But we can also regulate the activity of those insulators by doing things such as methylating those sequences such that they cannot block the activity of enhancers. So the general message I want you to take away from this slide is that transcription in eukaryotic cells specifically, I'm talking about eukaryotes, is a very complex process. And when we talk about the regulation or gene expression or the different mechanisms that a cell uses to do so, you will find that we will study a lot of these mechanisms at length, and you'll find that it's a very complicated, a very intricate set of mechanisms. So we're going to move on.